Okay, hello everybody. Today we're going to take a journey with FDA, with clinical research, in order to execute, run, and finalize a protocol. We're going to talk about working with FDA on how to optimize drug, device, and diagnostic development, and how working with FDA we get to the execution and completion of clinical trials. Next slide, please. So we're going to talk a little bit about, we're going to start from the beginning, and rather than just jump into clinical trials, so we can tell the story of drug and also device development. It's all related. So the first part of the drug development or device development is discovery and development, and that is somebody has an idea, they've come up with whether it's through their own research, whether it's through government research, however it happens, and they do actually the hardest part of the whole development process, and is they have an idea and they discover something. And once they discover something, which is the very creative part, now you have to go through development, and sort of it's like designing a bridge and then building it, being the engineer. So one is being the architect and one is being the engineer. So the first thing you have to do is you have to look at some preclinical work, whether it's be in vitro studies, animal studies, before we go into humans, so we can actually verify the concept that we're studying. The next part we do is, once we have this concept, we'll meet with FDA, which we'll go into a little bit later. And then we do clinical research, and we'll go through all the different phases of clinical research later. But there's a whole process, very well thought through and with a lot of experience, okay? Then ultimately we have the FDA review process, which is really the whole idea of having the regulators buy into our development plan. And by having the FDA as part of the development team, we mitigate the risk that at the end of the whole process, the FDA will not approve us. And then of course, once we have the approval, we have to do post-marketing surveillance to make sure that in the larger populations that we have a safe product. Next, please. So the general approach, because I think the philosophy is also very important. And the first part is we have to have good medicine. So there has to be a rationale of how we're going to help people, okay? And that's really very, 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 very important, because we have to think about all of that and how we're going to actually help people in the process, okay? The other is to have really good science, as we mentioned earlier, based on the scientific rationale. Most products are based on good science, but every once in a while, and even some of these things coming out of traditional Chinese medicine, just empirically, they have found that certain botanical products work. They may not even know the mechanism, but it sort of have experience. The other is we have to have good regulations. And this is really important, because our end game of all this whole process is to get the regulators, whether the FDA, the EMA, the Korean regulators, the Japanese, the Chinese, whatever it is, we have to conform with their specifications. And I think the other thing is, I like the idea of having passion and having pride in my product, because if you have pride in your product and you believe in your product, you'll be able to, first of all, be flexible in your development and the way you approach it. And the regulators and the clinical research sites will say, wow, these people really believe in what they have. Let's work with them. Because I think that's part of it. It's not just a business, let's get a product to the market and we're done. Every project that I've worked on, virtually every project, the people in the companies working on these projects all are very excited about what they're doing, and the goal is to get their product to the market. Next, please. So here's the other thing, is the team. It's really important that we don't isolate members of the team, but all the team is part of the whole process to the approval and beyond. So as we mentioned earlier, you have the drug discovery and development people who are usually the basic scientists or the engineers or the GMP facility, developing new procedures on how to develop and make your medicinal product. The other very important group to have is marketing people, because ultimately their goal is to sell it. But again, we don't want to have the marketing people drive the development process. On the other hand, we also don't want the scientists to drive it. There's a lot of the things that we do in drug development and device development are counterintuitive to what the scientists want to do. The other, actually, the key part is the clinical people. Clearly, we have to run the clinical trials and show that actually the product works and is safe in humans. The other is to make sure we have the legal people involved. That includes IP and includes all the patents, because we want to extend the lifespan and the life cycle of our product as long as possible. The other area, which is actually very, very important, is toxicology. And because there's two areas where you get into trouble. One, and usually the development people are the chemistry people, is either in the chemistry where there's some hiccup in the chemistry, or there's some flaw or some signal in the toxicology program that says we may have a problem, let's fix it. And the other members of the team are my regulatory group at my company who have to interact with the FDA. And what a lot of people don't realize, that the other key member of the team is the Food and Drug Administration. And the earlier we involve the Food and Drug Administration in the process, the more successful we will be. Next slide, please. Okay. And also we want to have a philosophy. Don't waste time, because time is money. And the business people will like that, because we really don't have the luxury of just thinking about things. And we have to plan carefully. And my philosophy is that for every hour we plan, we save 10 hours of chaos. Don't pin me down on the 10 hours. But as you know, there's some relationship of careful planning, so we don't have to do things too many times. Also, execute meticulously. Don't try to do short circuits and shortcuts in the development process. The other is, don't be afraid to replan. It's okay. In fact, FDA knows that hiccups occur during the development process. And the key is that we're not afraid to replan, okay, at all, okay? And if we need to, involve the FDA when you're doing the replanning. Also, especially during the early phases of development, only do what's needed, which means don't try to do too much at first. What I always tell folks is crawl before you walk and walk before you run. So here's an example I'll give you, which is now being sold by Colgate, and this is public information. We developed a product coming out of SUNY Stony Brook. First, it was a product to develop it as a prophy paste after you clean the teeth. The next, it was developed for tooth sensitivity, okay? And instead of calling it a toothpaste, we called it a dental cream, so it stayed as a device. And this product, under a very, very small budget, is now being sold by Colgate as a tooth-sensitive product. So, but we started first with what was easy to get approved, and that was just a simple prophy paste as a device, not as a toothpaste. And the other is hire people who want to get the job done. Zen Georgi, a Nobel Prize winner, said many years ago, if you want to get something done, hire someone who wants to do it, so that it's not just a job, okay? And we know at Target Health, it was a pride that we had people for 20 years at the company, and who always had passion, and who always were never afraid to say, I don't agree with what management is doing. I think we should reconsider, and management always listened. Next slide, please. So how do we prepare for the FDA meeting? And we're not going to focus a lot on this, but we have to tell a story. So what's our rationale for our drug? Any chemistry issues? Because almost always, separate from clinical, problems in chemistry are always on the critical path. So we think that this is something that's critical. It's more mechanical, operational, and it's not as, exciting perhaps as the clinical research, but chemistry is a big issue. And then of course the non-pharmacology and toxicology data, if we have any clinical data, and the key is to send a very good protocol and usually a clinical development plan. And could be the first protocol as a safety protocol, just the normal volunteers, which we'll get to later. And then we also give them a clinical development plan. Here's our concept. And if we have any publications, it really helps, because anything that we can get, we can supply in terms of peer-reviewed journals and things like that, becomes very, very, very important. Okay, very important. Okay. Okay, next, please. So what's in a protocol? So in order to execute the protocol, we have to write it. And so when you're writing this protocol, and there are templates on all of this, and a lot of these things are clearly defined in ICH, okay, which is a global group working to standardize clinical research among many geographical and political areas. So we have a synopsis, and sometimes with FDA, we can just send them the synopsis. Usually it's maybe a 6-7 page document, but it's pretty much detailed. What it doesn't have is all the boilerplates that you would have in a protocol, like data quality and stuff like that. But it has the guts of the protocol, including the objectives. Okay. What are we trying to accomplish? What's the study design? Okay. How is it a double-blind study? Is it an open label study? Is it randomized, etc.? All these specific areas. And very important is the exclusion criteria. Okay, which means, who are we going to include? And my general feeling is with inclusion criteria is the less difficult we make it, the better. So they should have the disease, males, females. If we're excluding females for some reason, we explain why. And we shouldn't just willy-nilly add inclusion or exclusion criteria just because we think it's a good idea. It should all be based on rational thinking. The other is a good description of the investigational product, whether it be a device, a drug, or a diagnostic. So it's very clear what it is. Is it a monoclonal antibody? Is it a small molecule? Is it a vaccine? Is it a dietary supplement? What is it, that we're actually studying? Then very important to study the efficacy and safety measures. What are the primary endpoints, secondary endpoints? And also in the initial protocol, we may find that once we get the study, we may change for the next study. So we may find some endpoint that we thought was really the right one wasn't. And that's part of the whole development process. The other is all the study procedures that we're going to do, visit 1, visit 2. Now with COVID, so many studies now are being done remotely. So we have to describe how we're going to do telemedicine. Are we going to send people to the home? We did a study in Gaucher disease where rather than having people come to get infusions at the office, we actually sent the phlebotomists and the people to the home so that we could do the procedure on the weekends. We could do it in the evening after work. So we were able to really include many, many more people. And this product was actually licensed to Pfizer. Okay. The other is statistics, which is also a very, very important part of the process. Okay. And because the other area that gets you in trouble, and the regulators are very serious about statistics, so don't treat it lightly. We actually had a program many, many, many years ago where for some reason there was 1 or 2 sentences that wasn't said correctly. We actually had to do the study over again, got the exact same results, but with the right statistical section. So we learned with this, and FDA, the statisticians have a lot of power in terms of shutting you down. And the other is run a high quality operation. Next. Okay, in terms of protocol execution, make sure the protocol is cleared by the FDA. It's usually the IND for drugs, an IDE for devices and diagnostics. Okay, we have to have the protocol and the informed consent cleared by the IRB or the ethics committee. Okay, and then we have to qualify the sites. Make sure the sites are qualified to do the study, and we'll get to a little more detail on that. And then we have to write contracts, especially in academic institutions, can take up to 3 months. And so that's also part of the critical path. So the quicker we can start the contract process, the better it is. And then we initiate the study, which is right before the study starts. We're ready to execute the study. We initiate it. We do training, okay? And actually, I was just involved with another study very recently, which I'm helping on fixing problems. They didn't do the proper training. So it was a study where they took the drug home with them. And when they had a flare of their disease, they would take the drug. But they found that the people that were trained intensively were much more compliant in the study than those who weren't. So don't minimize training, both of the patients as well as the sites. And then we screen the subjects and we enroll them, okay? Then we have a treatment phase where we go through the whole process of treating. And there's where the ECRF and remote monitoring becomes very important. And the more we can get the data entered in real time, the better it is. And then we can monitor in real time, we can monitor remotely. And if we enter data in real time, we don't have to worry about source document verification, which is a thing of the past. And then we eventually close out the study and write the study report. Next, please. Okay, so now we'll dig a little deeper into what all these things are. So what is site qualification? Okay, what does it really mean? Well, just because a site is a big academic institution or they're very well known, doesn't mean they can execute our study. Okay, so we want to really screen them and make sure that unless we want to just hire them for their name, but can they really get the study done? Because we learned in clinical research that often if we have maybe 20-30% of the sites that we actually enroll most of the patients, just the nature of the beast. Okay, so the more we can get the sites to make sure that they can execute the study, first of all, the quicker the study will get done and the more efficient it'll get done. We have to also confirm that they have experience in the indication under study and that the personnel have the quality, they have experience, and it's not just talk. We have to confirm the enrollment rate. How many patients do you think you can enroll every month, every week, every day? It really depends on the study. And it's also very, very important to identify any conflicting studies. Is there another study going on in oncology that has the same indication? Or another study that's going on in immunology or in rheumatology that's going to conflict? Now, sometimes it doesn't conflict because our exclusion criteria are very different. Let's say another study is doing males, but we're doing females. So there's no conflict, but there may be a conflict of resources. So we need to find all of that out. Next slide, please. The next is to look at phases of development. So in most cases, and this is in drugs, okay? It's a little different in devices. Okay. Sorry, there was some noise outside, so I tried to close the window. So in phase one in drugs, again, we're not, and it's different whether we're doing oncology, whether we're doing normal, and depending on or normal volunteers. But often what we'll do in phase one, we'll do pharmacokinetics, which means the distribution of the drug over time and its metabolites, looking at blood levels, looking at perhaps what's in the urine, perhaps what's in the feces, depending on the study. We clearly look at safety, so we're doing a lot of laboratory tests, electrocardiograms, other depending on the indication for use, electroencephalograms if we're doing neurology studies. And then we also do single ascending doses, so we want to pick a dose. So we do single doses, sometimes in the same patient, sometimes in different patients. It really depends. So all of these design issues come up, and we call a MAD study, which was multiple ascending doses. And let's say we decide to do at least a 1 week or a 2 week study in normal volunteers, just to look and see if there's any dose accumulation, whether or not we have any safety issues, looking at liver function, kidney function, other areas. And usually we have signals from the toxicology study what we need to look at. And those are normally normal volunteers. In oncology studies, the phase 1 studies, usually in patients. And usually we do people who have failed previous treatment, or the current therapy is not quite as good as they would like. And sometimes we'll actually take our product and put it on top of standard of care. Mostly that's in oncology, so it's a little different. So it really depends on the disease, and also whether or not it's an orphan disease, how we actually do the studies. Because for Gaucher disease, and this again, public information, we got approval with 30 patients, okay? Because there was an identifiable endpoint. We didn't do placebo because it would have been unethical. And we did 2 doses because there was a concept in the industry that maybe the current dose that everybody was getting was perhaps too high, or they could go on a lower dose. And we found that the lower dose also worked, okay? And again, we do dose selection and safety. And in phase 3 is the confirmation of effectiveness and safety in larger number of subjects. It really depends. And these are the kind of things we negotiate with the FDA. We have to negotiate with the statisticians, with power studies and power calculations, and et cetera, and things like that. Okay, next slide, please. In terms of phase 1, and now we're drilling down a little more detail. So it's usually performed in professional phase 1 clinics. And again, if it's oncology, it's often in an oncology clinics or an oncology medical center. That's like an MD Anderson, Roswell Park, and places like that. We normally will do normal healthy volunteers, so they're not on other drugs, they're not even on dietary. Now with dietary supplements being so powerful, we try to eliminate that. And also clearly patients, if it's oncology, or there's some reason not to do normal volunteers. I actually like the idea of going in to patients as quickly as possible, because sometimes patients are normal. In fact, we have one case where basically the patients were effectively normal volunteers because everything else was normal about it except they had the disease, but they were walking around and relatively healthy. We also want to always set up a safety committee. It can be called the Data Safety Monitoring Committee, whatever you want to call it, to review the data. Usually these are independent people, clinician, could be a statistician, could be an ethicist, okay, to look at the data. And in the protocol, we define when they look at the data. And there are stopping rules often, depending on, especially in oncology, where if there's, 2 SAEs, serious adverse events, we may stop the study, okay? Then usually the initial doses are based on the animal toxicology and even some of the pharmacology studies, because there's usually a ratio. We're not going to get into the details of the animal, the dosing and the animal toxicity. It usually is thought at least 10 times less than what we saw in the animals. And then we do our ascending single dose with PK and safety, and then multiple ascending dose. Next, please. Thank you. In phase 2, we always perform these in patients with the disease under study, and it's critical. We can decide whether or not we're using people who failed previous treatments, whether or not it's ethical to take them off their current medications. We just don't know. There's also a safety committee, clearly always, and when we're doing these studies. And the initial doses that we use are based on the phase 1 study for safety. Okay, because again, usually unless we have a biomarker indication, let's say like in phase 1, we could look at cholesterol levels or even blood sugar levels or some biomarker of disease or C-reactive protein. Usually it's just based on safety. And then the dosing frequency, do we dose once a day, twice a day, usually 4 times a day, is not something that's feasible right now, is based on the drug's half-life. And usually what's interesting is that in orphan diseases or rare diseases, we can often use adaptive designs. And the whole concept of this presentation today is letting you know that think about these things. Don't be rigid, and never, ever be afraid to ask FDA. One of the things I learned a long time ago with a colleague from FDA, he told me this was the head of the ophthalmology division. He said to me, never, ever be afraid to ask regulators if you have an idea that's even out off, out of the design, the normal designs, don't ever be afraid to ask. Worst comes to worst, they won't agree with you. Okay. And he was the kind of guy that he said to me also, if I don't answer your email, I don't answer the phone in one day, I'm probably dead. Okay. And when I did a presentation with him with an RX OTC switch that I was doing after I left Pfizer, and I won't go into the details. And when I said, I'd like to do this model, the primary study in a model system that you and somebody else published. And he said, of course, do it in the model system. This was allergic eyes, okay? Where we actually put the allergen into the eye and then we put the drug in, okay? And he said, yes, but then also I want you to do 50 patients who have the disease. So never be afraid to ask, okay? And so especially with the orphan diseases, there's a lot of flexibility and don't try to do too much. Next, please. And in phase 3, okay, what we do in phase 3 is we, first of all, we want to have a meeting with FDA before we start phase 3, okay, to make sure they agree with the patient numbers, the protocol, the statistical design, okay? Always, again, with the patients with the disease. And again, these could be more serious diseases. We have to decide early disease, late disease. In oncology, it really depends, and if we can actually help people who are dying, it's wonderful. Again, we have another safety committee. Again, the relationship from phase 2, we dosing, both the concentration and the frequency of dosing is based on phase 2. And I like the idea of doing more than one dose in phase 3, okay? And also we can do placebo if it's ethical, or we can even do it on top of standard of care. So the other thing is if you do it on top of standard of care, you may need more patients. Also, you may be able to replace standard of care. And you also should always have a rescue. So if for some reason your placebo is not working, we did this with head lice many years ago. If you had head lice, you had to be treated, but we had placebo, okay? So what we did is we brought them in, the patients in the next day and the families. Because with head lice, you also had to treat the families, okay? So we brought them in, and if the next day they had head lice, we treated them with the drug, okay? They were treatment failure, but we treated them with the drug, and then we brought them in the next day, and if they still had head lice, which was very few, it was a full treatment failure. We actually almost doubled the number of patients in the study because we had the rescue, okay? And often with phase 3, it's a multi-center and large numbers of subjects. Next slide, please. Orphan diseases, and again, these last couple of slides is thinking out of the box. You can use a lot smaller number of patients. You can also get in with FDA to buy in with accelerated approval, okay? And we were doing this with a company that had a biomarker, basically a surrogate, what's called the surrogate endpoint. And they were looking at something like glomerular filtration rate, which means how well the kidney is functioning, and do you improve it? So while it wasn't looking at kidney failure, looked at a biomarker that if you can actually improve kidney function, what they would do is they would measure kidney function, and they also did it with standard of care, or they measured with people under current therapy, and then they randomized them to their drug and saw if they improved, okay? Sometimes phase 2 is enough for orphan diseases, and then what you can often do is set up a long-term safety via a patient registry, and sometimes you can even do a study within a patient registry, and I actually published a paper with FDA and the Clinical Trials Transformation Initiative, which is a public-private partnership with Duke and FDA, on the role of patient registries in clinical trials. In fact, the VA, the Veterans Administration, you can do clinical trials within their database, because all they do is they have a question, are you in a clinical trial? Something like this, not to take me literally. And if yes, what is the treatment? So if you can have a separate randomization schedule, they treat them, let's say, with a drug A for diabetes or drug B or placebo, and then they just follow them the way they normally follow them in their standard of care. And again, always you'll have a safety committee. Next, please. Okay, and we also have to get FDA to buy in, okay? And one of the things we have to get FDA to buy in, and we brought up the idea of foreign data because part of this presentation is to our friends outside of the US. We have to make sure everything's made under good manufacturing practices. Hopefully the people making the drug are also registered with FDA. I think that's a really good idea. Get FDA to buy in on the use of foreign data. There's a whole guidance document on foreign data. So it's not an issue. Sometimes there are cultural or even dietary differences within societies. So FDA may want a local study, let's say in the US, but it could be supportive. So maybe we do one study in the US, but use the foreign data to support approval, okay? You can, if we're doing a foreign study and you have an open IND, we recommend you share that protocol with FDA, let them know what you're doing, even ask them their opinion. Again, outside of the US, they may not care. You don't have to use FDA requirements, but at minimum, we'll use it for safety. Always make sure your study is under ICH or International Conference on Harmonization and good clinical practices. And again, always make sure there's a safety committee with minutes, okay? So foreign data can become very, very important in this global world we live in, for sure. Next. In summary, okay, and thank you for your attention. Plan very carefully, always plan. Get the regulators on your team very early. Be transparent with the regulators. I assure you, they will find out. If you're hiding anything, as it's happened in the US, they will find out. They'll either be a whistleblower, like we had recently with the formula shortages, or they'll just find it when they do the inspection. Also, don't be afraid to get the patient voice, especially with orphan diseases. What's important to the patient? What matters to them? And also understand that the path to approval may take longer than expected. And for the financial people, it actually may cost more than expected. Thank you very much for your time, and look forward. If you have any questions, please send it to the team and we'll gladly respond. Have a good evening and a good day, wherever you may be.